Okay, so let's play with our learning objectives from the last 2 days. So I wanted to make sure we got all the way through them before Juneteenth, so that you guys could really have a good handle on this before next week and before we play with Punnett squares on Friday. Okay, so the first learning objective for Unit 5, Section 1 is to differentiate the information held on two homologous chromosomes. So remember, homologous chromosomes, right, are going to have this one and this one. Our homologous chromosomes are going to have the same genes, but they could have different alleles. So maybe big A, little a, okay? Versus these two also are going to be homologous chromosomes to each other, and maybe they're big B, okay? So here are my two homologous chromosomes versus non-homologous chromosomes are going to have different genes. They're going to be non-homologous chromosomes, okay? And then be able to look at sister chromatids. So remember that sister chromatids are identical because they're made from the same original DNA molecule, right? So we're going to have two sister chromatids. And remember that they're identical. And how that compares to non-sister chromatids. So non-sisters are going to be found on different DNA chromosomes, okay? And they're going to be single DNA molecules on different chromosomes. So these are non-sisters. And non-sisters, remember, have the same genes, but they might have different alleles, okay? And you should be able to figure this out at any stage of the cell cycle. So if I give you instead something like this, let's say at any stage. Okay, let's do a hard stage then. So if I give you something like this, and it looks like I'm homozygous for the B. Okay, so you should be able to tell me like, okay, well these, that, and that are homologous. You should be able to tell me that these two are non-homologous, right? We're in anaphase 1, right? You should be able to tell me that, this one and this one, these two DNA molecules are sister chromatids, and that this DNA molecule and this DNA molecule are non-sisters. Okay, so you should be able to tell me that at any stage. So you kind of got the straightforward one, like they were in like metaphase 1, I think, or like metaphase 1 before on an exam, but you might get kind of a harder one like this. Or maybe I'll give you something in a gamete or something like that as well. So just keep that in mind. You should be able to relate the terms chromosome, DNA, molecule, locus, gene, and allele to each other. Okay, so remember that chromosomes can be either unreplicated or replicated, okay? Let's just throw on gene on there, right? So either unreplicated or replicated. These things are both chromosomes. So an unreplicated chromosome has one DNA molecule versus a replicated chromosome has two DNA molecules. Okay, before I keep going, I want to make sure this mic is on. Okay. So a replicated has two DNA molecules. Remember as well that here a locus is a specific location on a chromosome and a gene encodes for a protein, okay? So here I might have this whole region being a gene, okay? And here I have the A allele, okay? And the A allele, it could be just a single nucleotide change, remember, a single mutation between this chromosome and maybe its homologous chromosome, where here we've got this big A versus the little a. So these are two different alleles, okay? So two different alleles of that same gene. Also remember that we have hundreds or even thousands of genes per chromosome. Hundreds, sometimes thousands of genes on every chromosome. our chromosomes are different lengths. So some of them contain more genes than others. And there's a whole bunch of other things that make it so that some chromosomes have more than others. So remember that there's lots and lots and lots of genes on chromosomes, okay? Okay, so we did this one. The last one is use the terms heterozygous and homozygous to describe the alleles present for a given gene. So up here, look what I did. I did big A, big B, okay? So if I've got big A, little a, remember that's heterozygous. And if I've got big B, that's homozygous. Okay, so when we're looking at a diploid cell, if we've got two different alleles, that's hetero, meaning different, versus if we have the same allele, that's homo, meaning the same. Okay? All right, so I think that's it for that one. Yep. Okay. So next, let's look at meiosis and fertilization. So you should be able to draw a cell that contains two pairs of homologous chromosomes with the genotype, with any given genotype. Sorry, that's a double up. With any given genotype. Okay, so for example, big A, little a, big B, little b. Oops, those look like they're in different spots. Don't want that. Big B, little b, okay? And so this you want to be able to make sure that you can do it at G1, oops, okay. But then also at metaphase 1. So they've been replicated, so this would be at like G1, so at met1, A, little a. No, it's hard to read, so go back to our longer notes, okay? So met1. I also want you guys to be able to figure out if the cell is haploid or diploid. So this is going to be diploid because it has homologous chromosomes, right? It's going to have 4 chromosomes. They're replicated there. Up here they were 4 chromosomes, and it was also diploid because we had homologous chromosomes, right? Actually, this is too messy. Let's make this bigger. Let's go down below, make it bigger. Okay, too messy, right? So let's just do a quick review of this, right? Aha, nice, okay. Big A, big B, little b, right? This is at G1. I'm gonna ask myself, do I have homologous chromosomes? Here, you can see I've got 2 different chromosomes. In this case, they do contain different alleles, but they are 2 different chromosomes with the same genes, right? So here I have yes, so that means I'm diploid. I've got 4 chromosomes. I have 4 DNA molecules. Okay. Then after S phase, right? Let's go to metaphase 1. We'll just skip all the way to metaphase 1, right? I'm going to have my sister chromatids being identical, my homologous chromosomes on either side of that metaphase plate. I'm going to mix it up and go like that. Right? Here, I'm going to ask those same questions. So here I have homologous chromosomes. They're on either side of the metaphase plate, but they're both there. So that's going to tell me that they're diploid. Okay. I have 4 chromosomes still. They're replicated, but there's 4 of them. And I have 8 DNA molecules. After all of meiosis 1, including cytokinesis, I'm going to have 2 cells. Right? One is going to have the big A's. The other is going to have the little b's. And the little A's. The big B's. I like to put my genes in different locations just because it's easier for me to track. So next, I want to ask myself those questions again. So now I'm in metaphase 2. And if I look at a single cell, okay, are there homologous chromosomes in just one cell? They're sister chromatids, right? So you should be able to tell me whether or not they're sister chromatids and non-sister chromatids. Here I have sister chromatids, but I don't have I don't have any homologous chromosomes, so I'm haploid, right? And I have 1, 2, 3, 4 DNA molecules per cell, and I have 2 chromosomes. So note that my number of chromosomes halved, okay? And that's what matters. So think haploid, half the amount, okay? So at metaphase 2, I have that. Then these are all going to separate, right? Sister chromatids are going to end up pulling apart during meiosis 2, okay? So I'm going to have big A, little b, big A, little b, little a, big B, and little a, big B. Remember that this is not the only option that we have, okay? So this is after meiosis when we're in the gametes. Either sperm or egg will show the same thing, okay? So homologous chromosomes. So no, right? Do we have non-homologous? So that means haploid, right? Non-homologous, yes. Okay, so we're always going to have non-homologous. So here you have like chromosome 1 and chromosome 2, right? Both in this single gamete. Okay, we have 2 chromosomes still and 2 DNA molecules, right? Right. So that's all of this, right? It was too small. So you should be able to draw them and explain if the cell is haploid or diploid. And with this explanation, I want you to be able to think about homologous chromosomes. Okay? The number of molecules and the number of chromosomes at those stages. Next, be able to draw the fertilization between 2 gametes and illustrate the chromosome content of each gamete in the resulting zygote. So let's choose this one for our egg, okay? And let's have a sperm that has little a, little b, okay? So they're going to come together, and the resulting gamete, right, is going to have that big A, little b from mom, and the little a, little b from dad, okay? So this individual is going to be, remember, heterozygous for A and homozygous for B, right? Where there's actually little b. Okay. You should be able to ask these same questions that we did up here too. So I want you guys to be able to say like, okay, in the zygote, right? This is a zygote. This is a zygote, and we're going to have those same questions. So homologous chromosomes. Yeah, we've got 1 from mom and 1 from dad, right? So the zygote will be diploid, right? We're going to have non-homologous chromosomes too, right? Those 2. We're going to have 4 chromosomes and 4 DNA molecules. Remember as well, this is more like review from before. Remember that this single cell, right, will go through S phase, and then it will go through mitosis, right? So let's actually just do S phase first. Okay, all of those are going to split apart. We're going to go through mitosis, and I'm going to line us up on metaphase of mitosis, okay? So in metaphase of mitosis, I'm going to just do all black again. But from this gamete, okay? Maybe it'll be better if I do this. Oh. Right? We have stuff from mom and dad, but at this point it doesn't actually matter, okay? Everything's just going to line up on that metaphase plate, okay? Back. everything will line up on the metaphase plate at metaphase of mitosis. So I like to think of mitosis like it happens in mitosis. Okay, mitosis. Okay, so in metaphase of mitosis, the sister chromatids are going to separate. Currently, I have 4 replicated chromosomes and 8 DNA molecules. Okay, and I have homologous chromosomes, right? And these two are going to separate into 2 different cells, right? Big A, little b, little b, big A, little b, right? So after mitosis, I'll actually be a 2-cell zygote at this stage. 2-cell zygote, right? I'm going to be back to where I was up here, right when I was a first zygote, right? So I'm going to have homologous chromosomes for sure. Okay, I'm going to have non-homologous. Oh, this is going to be diploid, right? Make me diploid in all those cells. Non-homologous. That answer is always yes, okay? I'm going to have 4 chromosomes, and I'm going to have 4 DNA molecules. Oops, 4. And then this is going to happen again and again and again and again until we get the trillions of cells that are present in our body, okay? And if we think back to that example before that we had of like, think back to when we had the curly hair, right? And the brown hair, right? Those are both being expressed in the hair, right? But they're on different chromosomes, right? But these weren't the only ones around. So for Sophia's gametes, you guys also did, right? The eye color gene, right? So actually, let's not put them there. Let's put them on the curly one. Okay. You also had that eye color gene. So maybe it was on the same chromosome as the curly hair, right? Remember that the hair genes are present in every cell, right? But only expressed in hair, right? And it's because that's where the activators. Right, so review all of that with the activators and the switches and the promoters and all those things, okay? The I gene, right? I gene is also present in all cells, but only expressed in the eyes, right? And again, because of the activators. Both of these genes, or all 3 in this case, of these genes are going to be present in my toes as well, but I don't have the eye color gene being expressed on my toes, right? Okay. All right, so let's go back to our learning objectives. All right. Okay, so we definitely drew that out. Draw fertilization involving 2 gametes and illustrate the chromosome content of each gamete and the resulting zygote. We definitely did that. Explain the role of meiosis in generating genetic diversity in a population. Okay, we haven't done that yet. So here, one of the ways that I could have lined up on metaphase 1 was for the little b to line up with the big A, and the little a and the big B to line up. Remember that another option could have been where it had the big A's on the left and the big B's on the left, right? Little A's on the right, little B's on the right, okay? And this would have yielded gametes that were big A, big B, and little a, little b, right? And that's different than the gametes that we got. Actually, I want to change that to not be green because it's not the sperm. This was in the egg still. Okay. Okay, so I want you guys to consider how that, right, is different than the other options that we got. So here we had gotten big A, little b, and little a, big B, right? So by lining up in different ways in metaphase 1, we can get all these different gamete options. We can get 4 different gamete options when we have 2 genes. When we have more than 2 genes, then we keep on increasing, okay? And like factoring that in. So if we had, you know, if we had something like big A, big B, big C, little c, right? You would be able to get A, B, C, A, B, C, A, B, C, A, B, C, little a, big B, big C, little a, big B, little c, right? And you can keep on going. Little a, little b, big C, little a, little b, little c. I'm sure I'm missing one, right? 1, 2, 3, 4, 5, 6, 7, 8. Actually, I think that's 8 is correct in this case, okay? So we could have made all these different gametes. So you don't necessarily need to be able to come up with all of these, but if I give you a genotype, if I give you the genotype big A, big B, big C, little c, you should be able to tell me what is not an option. So we cannot get little b, okay? So like if I were to ask you like, is big A, little b, big C an option, right? You wouldn't want to tell me no because it's homozygous for the big B allele, okay? Okay, what's next here? So that's explaining the roles of genetic diversity. Relates to the terms meiosis, gametes, somatic cells, fertilization, and zygotes. So remember that the meiosis makes the gametes, that 2 gametes come together during fertilization, and that makes the zygote, and then the zygote becomes an entire body through mitosis. Actually, I like adding that in here, through mitosis to make all those somatic cells. Okay, and the somatic cells are the things that like make us be able to move and see and all the things. Okay. Next is to label the chromosomes and chromatids and determine if a cell is diploid or haploid based on the chromosomes at G1. Okay, I guess we kind of already did that. I'm going to check it off because we did it here, right? So we did it kind of up here in this one, right? But then we also were figuring out if they were haploid or diploid going through mitosis here. Also be thinking back to when we did meiosis here, and then also be thinking back to when we did this in mitosis too, okay? All right, so I think that is all of our learning objectives. I know this is a lot for today. You guys do have a homework to practice with. I know you have a day off tomorrow, and I do want you guys to use your day off tomorrow, okay? So try to get all of this done today, and then just let it percolate and think about it until tomorrow when we'll work some more on some Punnett squares. Remember, we already introduced them, so it should be a pretty easy day on Friday. So I know it's a hard Wednesday, but then you get a break and then an easier day on Friday. And this is, on Friday will be our last day of new content, and then you'll be working on your project that I'll introduce probably on Friday. I'll do a little video on it, okay? All right, I hope you all have a great day, and I will see you again on Friday. Bye.